get started. So chapter 24 and really chapter 23 also focus on the origins of life. So specifically, how did life originate on Earth? So one thing you definitely want to have down for the upcoming prep quiz, which is this Friday in a couple of days, is the order in which the first sort of self-replicating molecules, these protocells, originated. So basically, it started with the synthesis of these small organic molecules. And then these small molecules started joining with one another. And we'll walk through this in a little bit more detail, what I'm getting at. But basically, it's kind of the discovery of, or the creation of RNA, and then the packaging of these RNA molecules into kind of vesicles, the product of which is called a protocell. And then from there, the origin of self-replicating packets of these vesicles. And so to know these steps, you really want to know what conditions these first molecules were kind of first experiencing. So kind of what was the scenery in which these first molecules came to be. And so the idea here is that, atmosphere on this early Earth consisted of very little oxygen. So remember, this is before life. Plants aren't and bacteria aren't generating oxygen. So there's very little oxygen in the atmosphere. The atmosphere itself is really thick with water vapor. So the Earth is extremely hot, and all the water that is there is in this vapor form. And then you also have in the atmosphere all these chemicals released essentially by volcanic eruption. So that's where you start getting things like nitrogen and nitrogen oxides, as well as CO2 and carbon-based molecules like methane, as well as ammonium and hydrogen. So the Earth was extremely hot in the beginning of this environment, but as it cooled down, all that water vapor that was in the atmosphere started to condense. Instead of being this gaseous vapor, it started to liquefy and settle down, forming these pools and what eventually became oceans. And as for the hydrogen that was part of this early atmosphere, that essentially just evaporated into outer space. But as the Earth cooled down, we essentially got this scenario where we start to have these oceans, these salt waters that even to this day encompass most of the Earth. One thing to be aware of are some of the early experiments that kind of tested how life might have originated. So we can't ever say for certain how it originated, but we can do experiments that try to mimic these past environments and see how we can create kind of the building blocks of life. So one of the most famous ones was this Miller and Urey experiment done in 1953, which kind of showed that you can get organic molecules made in what's called this reducing atmosphere, these early Earth conditions. And you can get organic molecules created by starting off with purely what are called abiotic factors. So abiotic meaning just non-biological, which makes sense in this early Earth, since we're talking about, again, the origins of life, and there is no biological factor. Biology implies life. And so these experiments were done to kind of see how can we generate biological materials, even just these small building blocks, through abiotic synthesis. Another study you might want to be aware of are some of the more recent experiments, like the fact that amino acids, which are the building blocks of all life on Earth, amino acids can be made in sort of much greater quantities when we imagine these deep sea and volcanic vents that were common in an early Earth environment. I mean, we still see volcanoes now, and we still see these deep sea hot environments. And in this 2008 study, the authors essentially showed not only that amino acids can form in these environments, these hot environments, but also that they can result in much larger quantities than the conditions that 1953, that Miller and Urey experiment showed. So that's what both of these graphs are showing, just in terms of the number of amino acids that were created and the mass of amino acids. So in this more recent study, these researchers found that, in these deep sea vents and near volcanoes, you make a lot more of these amino acids. So perhaps because you make a lot more, maybe this is the more likely scenario that resulted in the origin of these amino acids, the building blocks of life. So this figure here is really just showing another one of these experiments, showing how specifically once you have RNA monomers, so these are the nucleotides of RNA, just like DNA has nucleotides, which are, you might remember from Bio 1, the A's, T's, C's, and G's. Likewise, RNA has, instead of T, it has U, so, uracil, so A, U, C, and G. But what this experiment showed is that once you have RNA monomers form, so, once you get this situation, which these previous studies were kind of showing, how do you get those monomers? Once you have the monomers, it's actually pretty straightforward to get these small chains of monomers, which is essentially all RNA is, these long chains of different RNA monomers. And they actually form pretty straightforward when you find a lot of these monomers on these concentrations of monomers form on sand, clay, or rock, which are all habitats that these RNA monomers would have typically experienced in early Earth. So what this study is basically showing is that once you get this scenario where you have a bunch of monomers sitting on a hot rock, they will tend to form these small chains, which is essentially just RNA. Now, from these short chain RNA strands that might have served as catalysts for different chemical reactions, you can get a situation arise where these chains essentially just get trapped in this membrane-like structure called a vesicle. So when you're thinking of it, you can think of it like oil in water. When you drop a drop of oil in water, it forms these globules or spheres because the fat of the oil doesn't want to mix with the water. And so you get these essentially cell-like spheres form. And so these oils, which can sometimes contain RNA in them, are essentially just a protocell. So in water, it's been shown that these vesicles can kind of spontaneously bud off, essentially mimicking the act of reproduction. So think of it, when you put a big globule of oil in the water and shake it up a bit, it splits off, right? That's essentially all these experiments were showing, is that these vesicles too can bud off of one another, mimicking reproduction as the RNA starts generating more and more copies of itself within a vesicle. So another thing about these lipid layers, which you can see one here, is that these vesicles are kind of protective for the RNA. It can kind of create and help maintain this mini internal environment where the RNA can function properly. So what I'm describing is essentially just the most basic form of what a cell as we know it actually is. A cell, all it really is DNA within some vesicle, and it's a little bit more advanced than that, but in its most basic form, that's all a cell really is. Hence the name, in this case, protocell. And so by combining all these experimental evidences, researchers kind of showed how the right clay conditions can promote the production of these early vesicles. That's what's being shown here on the top. When you have kind of the right environment, these vesicles will just kind of increase almost exponentially before covering the whole clay area in these vesicles, which is kind of what you're seeing here. They basically have no more space to occupy, and so it just flatlines. And so basically over time on these ideal conditions, these clay environments, the vesicles themselves will kind of bud off of one another, forming more and more vesicles, which again, remember, that's basically a pre-reproduction event. And what they also found is that these vesicles can preferentially absorb RNA, creating essentially that protocell. So if they kind of eat up this RNA and bud off while the RNA is replicating itself within, that's essentially all cell division is, and reproduction is the production of more of the same genetic code budding off multiple and multiple times. So one of the big conclusions of this experiment, and others like it, is that the first genetic material, you heard me talk a lot about RNA, and from these experiments, it's hypothesized that the first genetic material probably was most likely RNA, not DNA. From this genetic material, and importantly, variation in this genetic material, meaning different RNA strands, you kind of get the blueprint for natural selection. And so following this line, you can kind of see the most basic interpretation of how we view natural selection. You know, vesicles containing RNA essentially produce quote unquote offspring that's similar to itself. And so from there, DNA might have arisen simply due to its kind of more stable nature. So one thing about RNA in general is that RNA is a very fragile, fickle molecule. DNA would have been advantageous in this environment, essentially allowing protocells to last longer and withstand different environments. And you know, natural selection is all about reproduction, and in this environment, lasting longer and budding off more times over the same amount of time will essentially result in the proliferation of that material, which in this case might have been DNA. And then from this point, you know, basically the idea is that all complex forms of life kind of exploded into all the species of bacteria, plants, animals, and other kingdoms that we know so well. They basically all originated, you know, that we couldn't have had any of these without the origin of life, the creation of these quote unquote proto-cells. And so when I talk about the origins of life, I kind of like looking back at this table. Kind of every textbook in evolution and biology has this near the beginning of the textbook when you're talking about the origin of life. And I like looking back at it from time to time because it's always so easy to kind of forget the timeline of life on Earth. But also on top of that, this table gives you kind of a cool sense of how recent our own history, not only as humans, but also just how relatively recent even the history of animals in general is. So to kind of put it in perspective, Earth is over 4 billion years old. Now, over the geological time scale, prokaryotes, and actually these first cells that I just described over these first few minutes, actually appear pretty quickly in Earth's history, 3.5 billion years old. That's kind of the best estimate of when cells first arrived, which is, you know, that's within, you know, kind of the first 10 to 15% of Earth's history. So it's not that, you know, we've had life in an early Earth, essentially. Animals, though, don't appear on the flip side until the last 10 to 15% of Earth's history, basically 500 million years ago. So the first cells might have originated the first 10 to 15% of Earth's lifespan, but animals came 10 to 15% in the last 10 to 15% of Earth's history. Another interesting thing to note when you're looking at this is animals are older than plants, which maybe surprises some people sometimes, since we tend to think of animals as these more complex things, but animals actually came before the vascular plants that we know. Dinosaurs, they're cool, and they were alive for about 200 million years, so essentially still a blip in Earth's history. Flowers are a relatively recent thing. They're younger than most dinosaurs, which might surprise some people. And then, of course, mammals, you know, where, you know, all the cute animals are. Mammals entered the scene with the end of dinosaurs 50 to 65 million years ago. And then on top of that, primates appeared only 30 million years ago. And now we're talking about just the last 1% of Earth's history. That's where essentially mammals and primates come in, is the last 1%. And then on top of that, bipedal human ancestors. So, the most primitive thing you might call a human appears in the last 0.1% of Earth's history. And even in terms of animals, that's the last 1% of animal history. So, whenever we say humans are a very young species, that's kind of one way to think of that. We've only existed, not even, civilization or think anything like that, but just us as a species. We've only existed, for the last 1.1% of Earth's history. So chapters 23 and 24 focus on how life went from kind of these single simple molecules to the diverse array of species, encompassing all the kingdoms and domains that we see today. Chapter 20 focuses a lot, not necessarily on the origin of life, that early Earth period, but rather the relatedness of all life that we see today. And so because of this, logically, you can see kind of how these chapters are related. If you follow each species' relatedness and ancestry all the way back, you end up essentially at that origin of life, that first time point that eventually leads to all life on Earth. So long as you don't count the aliens that walk among us, which clearly come from the Andromeda galaxy and have their own origin of life separate from ours. But anyway, and so one important thing that phylogenetic research allows us to do is characterize how closely related different organisms are. And taxonomy, a key vocab word you want to know, is really the discipline related to classifying organisms. Think of it as a sort of tool used within phylogeny to group similar and dissimilar organisms. So when we talk about taxonomy, we often talk about what's called binomial nomenclature. It's kind of a long term, but you have all heard of it and experienced it. It's how all species are named. We're all, as humans, referred to as Homo sapiens, except for those aliens that walk among us. Fruit flies are all Drosophila melanogaster. All species essentially have one of these species names that follow this basic binomial nomenclature. Binomial here referring to the two-part format that you see in all scientific species names. So our name, Homo sapiens, the first is the genus name, Homo, and the second part is the species or the specific epithet, sapiens in our case. The first letter of the genus, Homo in our case, is capitalized, and then the entire species name, Homo sapiens, is italicized. And you would not capitalize the sapiens part of Homo sapiens, just the H in Homo. And then both parts together, that genus plus the species, makes the name, the species name, Homo sapiens. But one thing that you might not be as aware of is that, in reality, our name, our classification is much longer than just a genus and a species. So the fact that every species has this name according to genus and species starts to get at this idea of hierarchical classification. And you'll read about this in your book, or you already have read about this in your book. But basically, you can think of classification broken up into these tiers of similarity. So in the most basic, you have the domains, right? You have archaea, and the probacteria. Then within the kingdoms, you have things like animals and plants. Phylum, it breaks it up within animal a little bit more than that. And then you get more and more specific into these smaller and smaller branches as you go down the line. So one thing you might want to remember is kind of the order in which these kind of tiers, these levels appear. And everybody has kind of come up with their own mnemonics for remembering, which is bigger than which. One you often see is Dear King Philip came over for good spaghetti. Basically, domain, dear, is the most basic classification. Then within domain, there's kingdom, phylum, class, order, family, genus, species. And so each one of these is a taxon. So a taxon is basically any group name. It can apply to the kingdom Animalia. It can apply to the genus Homo or the species Sapiens. These are all different taxa, which is the plural of the singular taxon. Now visually, you can also look at it this way, where domain again is the most basic. You have eukarya, bacteria, and archaea. Within eukarya, you might have the animals as opposed to the plants, fungi, protists, etc. Within animal, you could have chordates, which are anything forming a nuchal cord, like a lot of vertebrate animals. Within that, the class of Mammalia. This is all the mammals that exist on Earth. Then the order Carnivora, which is most of the carnivores on Earth. Within that, Felidae, which are the feline animals. Within that, potentially Panthera, and then that would be the genus Panther, and then Panthera pardus, the spotted leopard. So if you didn't like my mnemonic that I showed you before, you can learn a different one. Did King Philip cry out for goodness sake? For some reason, most of them involve King Philip. Don't ask me why, I don't know who he is. Now, because organisms can be classified and lumped into groups, this grouping system naturally kind of creates these branches where every twig within a branch kind of makes up its own phylogeny. And so because of this, taxonomy, classification, and phylogeny all kind of naturally go hand in hand. And you can see this when you look at these kind of well-made phylogenies, these phylogenetic trees. Each individual tip of a branch here might be an individual species, as you see here. And perhaps the first connection between two species might be all the species within a given genus. In the case here of the Canis genus, which includes coyotes, wolves, as well as dogs, which aren't shown in this phylogenetic tree. You can go back further and you find all the connections within the family Canidae. And maybe you have all members of the order Carnivora, et cetera, et cetera. You can keep going back until eventually, in theory, you could have the whole of life in one big phylogenetic tree. One thing to note is taxonomists often make mistakes. So phylogenetic trees and phylogenies are actually a dynamic field. As we gain more data and evidence, we correct phylogenies and get a clearer picture of how different organisms are related. It used to be that phylogenetic trees were based on morphological similarities. But since genetic sequencing data has become more and more accessible, there's been more and more of a movement to move entirely to these evolutionary relationships, looking specifically at constructing phylogenetic trees based on genetic sequence data and not just for morphology. So looking back on some terms that you might be familiar with, cases of convergent evolution might be cases where basing a phylogenetic tree on morphology might throw in some hiccups, right? So that example of the sugar glider and the flying squirrel, they look really similar, but genetically speaking, they're very distantly related. So if you build a phylogenetic tree based on morphology, the sugar glider and the flying squirrel are probably placed right next to each other. But genetically, we know that this is actually just a case of convergent evolution. They're very distantly related, genetically speaking. And so on a phylogenetic tree, based on genetic data, they'd be very far apart. So when you look at the phylogeny section in your textbook, you're going to see a lot of kind of overlapping vocabulary terms pop up that are also included in the reading guide. So remember, that's due Friday, which is when the next prep quiz on this on this material that I'm covering here is. So for the prep quiz, you're going to want to be sure of how to distinguish between these terms. But it's pretty straightforward if you kind of look at each term closely and break it down. So phylogeny is kind of describing that evolutionary history of a group of organisms. So you can kind of think of it as phylogenies and phylogenetics are the field that describe the evolutionary history of groups of organisms. A phylogenetic tree is going to describe that actual figure you're looking at, that thing that looks like a tree. When you see a figure, that's the phylogenetic tree. Phylogenetic inference is actually how we essentially construct that tree. It's how we assume different groups of organisms evolved based on that tree that we generate. And so we make phylogenetic inferences to create a phylogenetic tree, which describes the phylogeny that we're kind of hypothesizing. And then even further, that phylogenetic inference is based on what are called characters. So you can think of these as kind of individual pieces of evidence. So if we were doing morphology, before we looked at genetic sequence data and we look again at that sugar glider and the flying squirrel example, each morphological similarity kind of helps you to determine the proximity of two organisms on a phylogenetic tree. So we use these characters, and based on organisms looking more or less similar, we infer their phylogeny and place them closer along a phylogenetic tree. I hope that kind of helps clear these terminologies up. So just continuing with this, a phylogenetic tree essentially is a scientist's hypothesis about how groups of organisms are related. Each branch point kind of represents the divergence, so the divergent evolution of two groups of organisms from a common ancestor. And so sometimes you'll hear the term sister taxa come up, and all sister taxa are groups that kind of share an immediate common ancestor that is unique to that pairing. So just as an example here, glass lizards and monitor lizards in this phylogeny would be called a sister taxa because they share a common ancestor that isn't shared with any other species here. None of these endpoints go through this kind of node that we see. And kind of this is reiterating this difference between evolutionary history and morphology, this point that phylogenetic trees show patterns of descent, not phenotypic similarity. I think I've kind of hammered that point in with the sugar glider versus the flying squirrel point. So what can we and what can't we learn from phylogenies? One thing to note about phylogenies is that most phylogenies don't generally indicate when a species evolved. So, when you're looking at these phylogenies, it's not telling you the age of a certain taxa or a certain species, and it isn't telling you even how much change occurred in a lineage. So this isn't telling you, what percent humans and chimps share. There are phylogenies that do this, but we'll kind of cover those either in later topics and then in more depth in kind of upper level courses where you can estimate this. But when you see these phylogenies, you're not getting information on when these species evolved or how far apart they are from like a percent genetic similarity standpoint. And then one important point to make when we're talking about our own evolution, you often hear people say, a lot of people who don't believe in evolution, they say, I didn't evolve from any chimp, or I didn't evolve from a monkey. And this is true. Scientists don't think that either, though. What scientists believe is that humans and chimps evolved from what we call a similar or a common ancestor. So think of it as, some primitive primate here that is neither a human nor a chimp. And in one population, that population kind of experienced one evolutionary history that over time resulted in this chimp lineage. Meanwhile, a different population, ours, experienced a completely different evolutionary history that eventually led to the evolution of Homo sapiens, modern day humans. So this is kind of one important point to make that's often mistaken. When we say that we evolved from chimps, we didn't. We evolved from a common ancestor of other species, and you can take it back all the way to the origin of life. So I'll end this here since we're at about half an hour, but these are kind of some of the main points. There are others, you're still responsible for the reading and the reading guide itself.